Okay, we'll take questions. I'm going to begin with Sneha. Sneha, go for it.
Sir, I have a doubt. In smooth endoplasmic reticulum, it is rich in enzyme. So, can you tell which enzymes are rich in?
Okay, there are many enzymes that are present. If you really want to know, just go look up. I'm not discussing any specifics here. Okay, any standard textbook, if you pick up, it'll tell you all the different enzymes that are present there. Some of the enzymes we do know, and we've characterized them. Many of them are enzymes that actually synthesize lipids, for example. Some might be those that actually modify proteins in ways that allow for them to now be integrated into the membrane or bound to the membrane, allowing for their delivery, right? So I'm not getting into specifics on any of these, but you're welcome to go look this up, right? And the textbooks that you have will tell you a little bit more about what enzymes and what exactly they do, right?
Okay, thank you.
Vaishnavi, next question, over to you.
Sir, different organisms with different requirements would have different Golgi apparatus, or the contents would vary, right? Would they?
So could you say that again? Different organisms as in?
Uh, from unicellular to multicellular, would their requirements change? So would the contents of the Golgi change?
At a single cell level, right? The functionality of many of these components is conserved. But depending upon the type of cell that you're looking at, right? And the kind of proteins that are being processed. Like, for example, the Golgi in a neuron may have slightly different composition, proteins that it is seeing, enzymes that, it uses as compared to Golgi, say, in the liver, right? So there will be differences depending upon which cell type you're looking at. A significant part of the architecture and the mechanisms that are in place are conserved. But the way these mechanisms are used, what kind of proteins are being processed will obviously be different.
Okay, sir.
Okay. Varad.
Sir, I didn't understand the when you were saying that the forward and the backward movement of protein through the Golgi. What were you really saying? I didn't understand.
Right. So this is again, something that is hypothesized. We don't fully know whether this actually happens. See, a challenge with a lot of this has been being able to image these structures. So to be able to look at a Golgi or an ER, as much as, I showed you images of confocal images, you can see their architecture to a certain extent. If you really want to see the stacks of the Golgi, you will have to go to something like an electron microscope, right? And one of the challenges with electron microscopy initially, the challenge was that you were able to look at only one plane. But now there is EM tomography, right? Where you do confocal sections, you can do sections in an EM image and also put the sections together. So the ER image that you saw of, the parking lot image, right, is made possible because they were able to process a stack of electron microscopy images. The challenge, so the EM tomography has allowed us to see these architectures a little better than we originally did. But the challenge still remains that we can't look at live events as they are happening in EM. Right. So it is hypothesized that there are vesicles that are being moved to the front, and they go from, as I said, one of the hypotheses is that everything just goes through, right? And one hypothesis is that there are actually vesicles that are peeled off from one, go to the next, go to the next, go to the next. And it is thought that there are certain vesicles that may also move back. For example, if and how this is controlled, what is the mechanism, we don't know. What decides that something should move forward or go back, we don't know, right? So it's thought that the way they are moved back and front will determine the residency time, right? That means how long a protein that's in this vesicle stays in this compartment could be determined by how quickly it leaves and how often it comes back, right? So what if there is a forward back that is constantly happening, right? And if all the processing that a protein needs to undergo in this compartment has been done, then it moves forward and then doesn't come back and then moves further. Could that be a way of doing this and ensuring that all the changes that are required are actually have happened on the protein before it actually leaves? So one of the questions here to think about is, what is the regulatory mechanism that looks at a protein? What is the quality control mechanism, right? That looks at a protein and say, okay, all modifications are done. Isko jaane do aage, right? Or looks at everything and says, "Arey, this particular modification has still not been done. We should send it back to that compartment." Is there such a a mechanism to check for the modifications that proteins are undergoing? And we actually don't know at this point of time. We are only just beginning to understand how changes can happen. Simple things like the amount of time that a protein spends in the compartment could change the architecture of the modifications, right? So a protein that will take, I'm just saying these numbers off the top of my head, that takes 30 seconds, 30 seconds, 30 seconds will be modified one way. A protein that takes 1 minute, 1 minute, 1 minute, 1 minute, same protein will be modified differently, right? This we've understood, or at least we've been able to now model, use mathematical models to suggest this is possible. And this has happened in the last maybe 5 years or so, right? So we're just beginning to understand the complexity and the kind of regulatory mechanisms that exist here. Okay, so I know this isn't much of an answer, but that's where the understanding of the field lies at this point of time, right? Aditya, next question from you.
Sir, can you hear me? Like neurons are really big cells, right? And they have many synapses.
So they will have one synapse. They don't make multiple synapses.
No, like at the end, let's
Yes.
say,
Yes.
they have only one synapse.
Yes.
Also, does the synapse have a broken part, like a piece of Golgi or something, a localized Golgi, so that it's easier for processing to happen before it's released?
Yeah, so whether there is a piece of a Golgi that sits right at the end, I don't think there is, right? So the Golgi is here. Actually, these motor proteins they travel long distances in the neuron to actually take things all the way to from one end where they are made to the end that they are actually delivered. So, so the way motor proteins work in neurons now is the basis of very extensive studies to essentially characterize this, right? You should look up, there is a lab in IIT Bombay, Sandhya Kaushika. Sandhya studies these motor proteins, and she studies how in cytoskeletal components in neurons, things are actually moved. How do they bump into each other? What is the level of crowding? How does such a long cell navigate, and carry information, right? Because, halfway through, what if the vesicle forgets where it's going? Does that really happen, right? And how does the neuron maintain that because of the distances it has to travel, right? So the short answer is there isn't a Golgi-like structure at the end. Things move actually long distances in the neuron.
Okay, sir.
Anjali, your query.
Sir, what do we actually mean by processing of the materials in the Golgi apparatus? Like, what essentially does the Golgi apparatus do?
Right. So we saw, right, one of the big processings that they do is glycosylation.
Sugar addition.
Right. Yeah. Glycosylation. There could be post-translational modifications, right? Like, for example, in some cases, proteins need to have lipid modifications, right? And this allows, there are, palmitylations, it's essentially a chain of lipid that gets attached to the protein at a certain point that now allows the protein to anchor to specific compartments in the cell using that lipid tail. Those kind of modifications can happen in the Golgi. There could be phosphorylation of a protein that allows it to kind of go to a specific compartment or bind something, which could happen at the Golgi. So many of the changes are actually at the level of the structure of the protein, in the way the protein is put together. Changes like phosphorylation, which are more transient, right? That can happen quickly and go away quickly, happen after all this processing is done. So glycosylation, for example, is a slow modification and doesn't kind of go away, right? So the changes that happen in the Golgi kind of stay, right? And stay longer. And those are the kind of changes that happen at the level of the Golgi. So there are many such modifications of the protein which affect the structure and functioning of the protein that happen at the Golgi.
Also, sir, when we said that we detach the Golgi apparatus, it becomes spherical rather than the normal structure. I didn't understand
Detach
that
the
part.
Golgi apparatus? No, that's not the Golgi, that's the cell. We are detaching the cell.
Okay,
Right?
yeah.
And when we detach the cell, the Golgi breaks up. Okay, that's what I showed. So what was inside the cell is what is the Golgi that is broken up. Sneha, your query.
So I have a doubt on smooth endoplasmic reticulum. So how does it catalyze it?
I don't know what you mean by how. These are enzymes. The enzymes, work on the proteins. Like just as, for example, enzymes that talk to proteins in the Golgi are like other enzymes. They will need a bunch of cofactors. In some cases, they may need energy. But when they run into a protein that they can modify, they will hold the protein and modify it, right? So a lot of that happens very intuitively. It requires a bunch of cofactors, bunch of other players that are required. A protein by itself may not exactly by itself be enough to do this, right? And so that milieu in the environment and what else is needed is actually vital for this. If you're curious, just go look up molecular biology of the cell and how Golgi or ER processing takes place, and there is a lot of information there on how enzymes, which enzymes and what kind of modifications they can create.
Yes, and so one more doubt. We said that Golgi spreads at a certain time. So does it affect the cell?
Spreads at a certain time, as in? Did you say spread?
Yes, sir. So as it moves away, as it and it catches up with the whole cell.
The Golgi catches up with the whole cell. No, I'm not sure I understood. Can you frame that question differently? Why don't you think about what you're asking, and I'll take the other two questions and I'll come back to you, Sneha.
Okay, sir.
Anandita.
Sir, so
yes.
Even endoplasmic reticulum can modify the protein, it's just not Golgi?
Yes. So there are some changes that can happen at the ER. There are some changes that can happen only exclusively in the Golgi. And the Golgi, now we understand, has a much more finessed, and complex mechanism than probably the ER. The ER does make changes. It's not like it doesn't. But there are some changes that can happen only in the Golgi, and the Golgi associated changes are more subtle in ways that allow or affect the functionality of the protein.
Okay, sir. Thank you.
Ajinkya. Okay, we have last two questions.
So I have one question. Does the Golgi apparatus change its shape for maturing different proteins?
So I'm not sure it changes its shape to allow for maturation. I think the change in shape might affect maturation. Okay, so I don't think the protein has a say in how the Golgi looks, but there are other things in the cell that will affect the organization of the Golgi, and that will mean that the protein can be modified this way. Okay.
Okay, so what decides whether, like, how long a protein stays in the Golgi apparatus?
We don't know actually. Right. So there could be some signature in proteins that could influence this. For example, if vesicles are thrown out and then brought back and then go out and then come back again, what is that mechanism? We really don't know. We don't know. Deep, last query.
Sir, you said that to study Golgi, we do not know how to take it apart, and like, study things separately. But sir, like in division, it is already taken apart now. So why don't we study the Golgi apparatus while it is dividing? Like,
Right.
there are,
The challenge with that is it's a very transient process. And the imaging capabilities that we have, actually a lot of understanding of how the Golgi is broken up, how it is brought back together, has come from the dividing cell. But the challenge has been being able to visualize things at that stage, and also being able to catch things as they happen in a dividing cell. Like we study the dividing cell in the lab, and just being able to watch the cell in a short period of time can be very challenging sometimes, right? So a lot of this is limited by actual data being generated, right? And then now asking, how do we look at the data and then analyze it? So challenge has been being able to actually experimentally do things here.
okay.
Sneha, come back to your question. Last question.
So I couldn't frame it properly,
Right.
but sir, in the slides, it was shown that it is moves throughout the cell.
It is what?
It moves throughout the cell.
Moves throughout the cell. So the Golgi can move around in the cell, right? The Golgi can open up, can come back together. In a migrating cell, for example, the Golgi, because it is tied to the microtubules, comes to the front of the cell as well, right? And when I say front, it's not right at the front, but if this is the direction the cell is going, right, the Golgi will be here and not somewhere there, right? That difference we have. So the Golgi can be organized and brought to very specific sites inside the cell to allow for it to support transport and delivery of vesicles and the components that the vesicles carry, right? So if that's the question, yes, it can be moved around inside the cell. Okay,
Okay, sir.